de donde es oriunda, y allí inició investigaciones en los bosques nativos de su país. Realizó un postdoctorado en Bangor, en Gales, y en Oxford, en Inglaterra, antes de incorporarse a St. Andrews, y a partir de ahí ha hecho una trayectoria dedicada al estudio de la biodiversidad, de la estructura de las comunidades y la ecología evolutiva. A lo largo de su carrera, Annie ha investigado plantas, invertebrados, vertebrados en ecosistemas de agua dulce, marinos y terrestres, trabajando en múltiples países y continentes. Si ustedes van a la página web de la universidad y buscan a Annie, van a ver el mapa, está por todo lado, pero le falta Antártica. Su investigación se centra en comprender cómo medir la biodiversidad, cómo varía en el espacio y en el tiempo, y qué procesos ecológicos modelan la estructura de las comunidades. En el contexto de la actual crisis ambiental que estamos teniendo, su enfoque adquiere una especial relevancia. A través del proyecto ERC Advanced Grant Biotime, su equipo ha mostrado que la principal amenaza de los ecosistemas no es únicamente la pérdida de especies, sino la reestructuración de las comunidades, con cambios en la composición, en la abundancia y también en la función, pese a que el número de especies siga constante. La producción científica de Annie es extraordinaria. Ha publicado más de 200 trabajos, entre artículos, capítulos de libros, monografías y contribuciones editoriales, así como 29 bases globales de biodiversidad. Sus investigaciones han recibido más de 80.000 citas a nivel global y por eso se posiciona como la segunda ecóloga más citada en el planeta. Además de su destacada labor académica, Annie es reconocida por su generosidad hacia los investigadores y las investigadoras jóvenes. Fomenta la interacción con su equipo, promueve la diversidad cultural y crea espacios de trabajo en los que todas las voces son escuchadas y son respetadas. La experiencia, cuando uno le pregunta a Annie, se nota la experiencia en la claridad metodológica con la que guía las respuestas. En Argentina ha colaborado con estudios sobre diversidad taxonómica y funcional de especies marinas, dejando influencia formativa en científicos y científicas locales. Por eso, después de esta charla, animo a los estudiantes y a las estudiantes para que se acerquen a Annie y le pregunten sobre sus análisis, sobre la expectativa que tienen como carrera, sobre los paths que pueden seguir. Ella siempre va a tener un consejo para dar. Agradecemos profundamente la voluntad de Annie de acompañarnos y de venir a Argentina por sus propios medios y de aceptar la invitación de la organización para estar acá en Madrid y acompañarnos hoy. De acompañarnos con su conocimiento transversal en biodiversidad, con toda una comunidad que está enfocada a estudiar el mar argentino. Gracias.
Buenos dias. Thank you, Patricia. And thank you very much to all of the organizers for inviting me here and to everybody for making me so welcome. Today, I'm going to tell you about some of the research I've been involved in biodiversity, which is one of the big challenges of our era, because of course we find ourselves in the midst of a biodiversity crisis. So what can we as scientists do to help understand what's happening and to help change the world become a better place? So the focus of my talk then is how and why are ecological communities changing over time? And I'll try and do that mostly in the context of marine systems. Now, if we are quantifying biodiversity, we need to think about the structure of ecological communities. And the conventional way to do that is to contrast biodiversity in particular areas, which we call alpha diversity. How much diversity do you find in a particular place and time? We might count species, for example. And then we might be interested as well in compositional differences between sites. And we typically call that beta diversity, or if you're American, beta diversity. And then we can think of the diversity of whole regions, and we normally call that gamma diversity. These ideas come from Whittaker, who was an American scientist in the 1960s. And we can think of that framework over space or over time. And it's a useful way to conceptualize variation in biodiversity. As well as that, we can think of the structure of assemblages. So we can just count species, but we might also be interested in the relative abundance of species. So we can think of that as a dial, and as we can change the weighting and use different metrics to emphasize how much of the structure of the assemblage we want to quantify. And this is often called, excuse me, they use, these days they're called Hill numbers. So we can think of richness as Q0, Shannon index, which weights by abundance a little bit more, as Q1, Simpson index, which weights much more by evenness. So we've got this structure in space and time, and we've also got the emphasis on how much we're interested in the relative abundance of species and assemblages. Now, there's a huge literature on how to measure biodiversity, but one of the more interesting and recent developments that I have been very slightly involved in is very much led by this lady, Ann Chow, who's a very clever statistical ecologist from Taiwan. And she's found a way of integrating all of these different approaches in the same statistical framework. And this makes it, provides a much more robust way of quantifying biodiversity and understanding biodiversity change. And I can illustrate that by taking you to Scotland. Obviously up here, St Andrews is just right here. But we're interested in this context in the west coast of Scotland. And these are data which were collected by scientific fish trawls over a number of decades. And if we examine those using the Chow framework, we can see that we get different signals of biodiversity change depending what metrics we use. And this is very important because it gives us an insight into how these systems are being reorganized over space and time. So here we've got Q0, which is species richness, Q1, which was Shannon index, Q2 with the Simpson index. We're comparing fish assemblages in the north and the south part of that region since 1990 to 2010. And we've got gamma diversity, alpha diversity, and beta diversity. And just by glancing at this, you can see that the different measures of biodiversity are doing different things. Some are rising. For example, compositional change is accelerating over time. Some are declining. Local alpha diversity and overall gamma diversity is showing a decline. And also we can see different strengths of signal in the north and south. So an integrated framework like this is a very powerful way of understanding spatial and temporal change in biodiversity. But of course, it's more than that, as you will have heard talking about other ways of measuring biodiversity. We can think of not just taxonomic diversity, what species there are, but also functional diversity and phylogenetic diversity. These are all measures of what diversity in a system is. And Ann Chow has also extended her framework to include these different dimensions of diversity as well. So again, we can use the same statistical approach to have an integrated understanding of variation over space and time. Now, I don't have a marine example of this extended framework, but I do have a terrestrial one, which I will use to illustrate it. So I take you to Scotland again, and these are pollen deposits, which have been put in place since the last ice age, 10,000 years ago. And you can use those to reconstruct the vegetation in the area. And you could do the same thing with cores from the ocean as well. And this is the result. So here we've got taxonomic, functional, and phylogenetic diversity. We've got two habitats, two areas, a blue one, a red one. We've got the length of the time series, 10,000 years before present. And we've got the three metrics of alpha diversity, species richness, Shannon, and Simpson. And what you can take away from this is that the different dimensions of diversity give us different signals of biodiversity change, and that the differences between the two communities vary depending on the weighting that we place on our measures of diversity and also dimensions of diversity that we use. This is just an illustration of how we can robustly quantify biodiversity over space and time. So different metrics provide different insights into how biodiversity is changing over time. And I think that helps us when we go out to study biodiversity to really articulate very carefully what questions we're asking, and then we can draw on the correct tools to try to answer that question. Now, everyone is aware that the world is in trouble and that biodiversity is under huge threat. So a big intellectual challenge at the moment, and practical challenge too, is to understand the changes in our contemporary world. How is biodiversity changing as a consequence of humans' activities in the environment today? And there are many, many different studies examining this. This was some work which led by one of my PhD students, which looked at the diversity of fisheries catches over the last 65 years. There's some nice data sets that you can examine. And so we've got bony fish and cartilaginous fish. We've got taxonomic richness and functional richness. And the color is the amount of increase in functional diversity extracted from the system over time. So the redder the areas, the bigger increase in functional diversity in the fisheries catches over the last 65 years. So clearly we are extracting a lot of diversity from the oceans in terms of our fisheries catches. Now, to examine these sorts of questions in a more systematic way, I think Patricia mentioned the ERC project Biotime, which I started 10 years ago, and it's been followed up by lots of my colleagues ever since. So we set out to try and quantify change in biodiversity in ecosystems across the Earth. And to do that, we started to compile the Biotime database. And this consists of ecological assemblages which have been sampled in a consistent way over a number of years. This was the initial study, and I seem to have lost the outline of the slide. But anyway, this is where we are here, roughly. And excuse me, these are the samples or the assemblages that we were able to compile. It's both terrestrial and marine. And there were 100 time series and over 35,000 species in total represented. So this is the first result that we got from that. So this is looking at change in species richness over time in these assemblages. So this is the year of the study. Each of these is a different assemblage. It's color-coded by taxon, and this is the common trend across all of these assemblages. So you can see in some cases richness increases, in some cases it decreases, but taken overall, there was no systematic trend in species richness, which was something which went against the common assumptions of the time. Biodiversity is in crisis. We must be losing species everywhere. But in fact, if we go out and measure the number of species in these assemblages, on average, there's no change. But what is more interesting is the second finding. And that is, if we look at how the composition is changing relative to the baseline, and here the baseline is just the first year of the study, we can see that the systems are becoming increasingly more dissimilar over time. This is a thing called a Jacquard measure, and as the way it's presented here, as lines go down, the assemblage is becoming more and more different from the baseline over time. And this is the common trend here, which slopes down gently. Now, we know ever since Darwin that composition does change over time. But what we did in this study was to compare that degree of change with the best ecological theory that existed at the time, and that included the Hubble nuclear model. And what these data were showing was that these assemblages are changing more over time than ecological theory would predict. So there's a big degree of compositional reorganization in these systems. When this paper came out first, it was very controversial because we said that species richness was, on average, not changing a lot. And people didn't really seem to notice so much the compositional change, but that's beginning to change. And now compositional reorganization is recognized to be a much more important aspect of biodiversity change. So elevated compositional change, temporal beta diversity, but no consistent change in assemblage size, temporal alpha diversity, what that study showed. Now, some follow-up work that has been done has looked at the extent of compositional change in different systems. And this study pulled together data from polar, temperate, and tropical regions across marine, terrestrial, and freshwater ecosystems. And this is plotting the degree of temporal turnover and compositional change. And I hope you can see that across all the systems, there seems to be more compositional change in marine systems than in terrestrial or freshwater ecosystems. So there's certainly a lot of compositional reorganization happening in the oceans, and it also includes a lot of different taxa. The zero line would be no change in any of these systems. Now, since then, my colleagues in particular have extended the Biotime database. It's published earlier this year, and it's accessible to anybody who wants to work with it. And so it's a compilation of time series in which you can do biodiversity analyses as you wish. And so this later version of the Biotime dataset is already being used quite extensively by many different studies. And this is one that I was not involved in, but it looked at turnover rates in different systems again. It's published in Nature earlier this year. And again, it reinforces the point that marine systems are changing much more in compositional reorganization than freshwater or terrestrial ones. And this particular paper relates that finding to the degree of warming or cooling. So climate change is being implicated in the degree of compositional reorganization in these systems. What I want to do now is to try and dig into these patterns a little bit and begin to unravel some of the processes that underpin them. So this was, I mentioned before that species richness was apparently fairly consistent over time in many systems. So in a study which was led by my colleague Nick Catelli, we had a look to see whether there is a signal of regulation of these systems. Is there some inherent property in the system that keeps assemblage properties more or less constant over time? And we call this regulation. And so what we mean by regulation, a regulated system looks like this. A random walk system might look like that, so not very different. But the criterion of a regulated system is that it comes back to a previous level after perturbation. So it looks like this rather than like that. And there's a statistical test thing called an augmented Dickey-Fuller test, which we used to test that. And what we found was, and this was tested on marine fish assemblages, most of what I'm talking about would be fish, and we found that the majority of systems showed this signal of regulation, statistical regulation. And these are the actual results. So anything to this side of the graph is a random walk. Anything to that side is a system showing this statistical regulation. And we find evidence of statistical regulation in terms of both species richness and total assemblage size in terms of total numerical abundance. So there are inherent properties in systems which seem to be keeping the assemblage properties on more or less the same level. So that begins to contribute to the finding that there's a lack of systematic change in many systems in species richness. The other thing that we find in a follow-up study, also led by Nick Catelli, is that a relatively, if we're thinking about temporal beta diversity now, compositional change, a relatively small number of species in the system are the ones which are driving the reorganization of community composition. So this is, again, draws on temperate marine fish assemblages, and this is looking at, is classifying the different species in the system into a range of temporal change types. So we have ones that are always there. We have ones that are there, but come and go systematically. They're ones that show random behavior. And so these together account for most of the species in these systems. And then we have a small fraction of species which are increasing or decreasing in particular ways. And these are the ones that are driving the change. So the message from this slide is that most of the species in the system are not contributing to change, but there are a small number of species which are driving the patterns of compositional change that we see. So, summarize that, temporal alpha diversity is regulated in many marine fish assemblages, but a small number of species are underpinning the changes in composition that we see. Now, to try and elucidate these findings a little bit further, I'm going to take you to a study that has been, I've been involved in for a number of years. And, excuse me, this involves a rather unusual sampling device. It's a nuclear power station in southern England. And you can see the water intake there. Managers of nuclear power stations don't particularly want fish into their turbines. So there are grills to catch the fish. And my colleague, Peter Henderson here, went to this nuclear power station every month for 40 years and collected the fish that were trapped on the intake during a particular phase of a tidal cycle and counted them. So it's a wonderful time series, and it gives us a great opportunity to disentangle some of the processes that I've been talking about. The first thing that Peter and I did was to look at the distribution of abundance over time. And what we found here was that we have some species that are there all the time, rather like the ones in the slide I showed you. So some species, this was a 30-year time series at that point, some species are there every year. Some species come in and out, like lights blinking on and off, and there are relatively few species of intermediate abundance. And we can represent the same data in the form of species abundance distribution. So these are the core species or persistent species. If you're interested in species abundance distribution, it's a nice classic log normal distribution. And the occasional or transient species are much less frequent, and they're more typical of a log series distribution. So this first study showed that temporal appearance in a system is linked in interesting ways with species abundance. We followed that up by examining the temporal performance behavior of these different species. And what we found in this study was that the core species, the persistent species, were largely showing density-dependent dynamics. So their abundances were pretty constant through time, and if the population declined, it would come back up to the previous level. we find that the transient species, the very rare species, were not under density dependence. Their dynamics were much more random. These were like lights blinking off and on in the system. But this interesting group of species, the ones that were there most of the time, I should say persistent or core species were ones that are there maybe 2/3 of the time series. So there are a number of species that were not under density dependence. And you can see the behavior of these on this slide as well. So these are the core species under density dependence. They have, they're the most abundant ones. The very rare ones which have vanished in this slide are the light gray ones. And it's the red ones, core species which are not under density dependence, which are the ones which are changing their abundance most through time. So these are the ones that are actually driving the compositional change, the species that are there most of the time, but are not showing very stable dynamics. And this sort of illustrates the same point. It's just variation through time, coefficient of variation versus variation in biomass. You can see the blue ones, the core density dependent species, very little variation through time. And these red ones are the ones that are driving compositional change. So it's these persistent or fairly abundant or fairly frequent species that are not under density dependent dynamics, which seem to be driving a lot of the compositional turnover in the system. Now, like many systems in the world, the Bristol Channel, the Irish Sea, which is reflected in these data, is undergoing environmental change. And these are just some plots to show it. So time, years, 40 years now. So we've got the mean annual temperature, degrees centigrade, increase, and also an increase in the resource base of the assemblage. So this is the biomass of the crustaceans in the system. And the crustaceans, the macrocrustaceans, are the ones that are fueling a lot of the fish community dynamics. However, if we start to look at change in the biodiversity metrics, it's not that dramatic. So here we've got species richness through the same 40 years. It's increasing slightly, but it's not significant. So it's nothing like the same increase as we're seeing with the environmental variables. So that leads to the question, which ecological mechanisms contribute to the regulation of these assemblage properties? And here I'm going to argue that ecological communities are subdivided into groups. And because the groups respond in different ways, they can average out the overall community response as a sort of form of what's called the portfolio effect. And if we go to this system, we can divide the fish community into 4 main groups. These are the pelagic fish. Well, people here will know this very well. One's proximobenthic that live close to the bottom. And in this system, we've got soft bottoms and hard bottoms. So we've got soft benthic and hard benthic groups of fish. So we call these spatial gills. We also need to think a little bit about how we measure the importance of species. So we can count it up by just counting individuals, numerical abundance, or we can actually measure the weight of the species in the form of biomass. And as you will well know, fish vary hugely in how big individuals are. And these are the 2 extreme species in the system, tiny goby versus a huge conger eel. So I'm going to think about these 4 spatial gills, and I'm also going to think a little bit about how we measure how abundant species are and ask how that contributes to the overall regulation of these trends. So here we are with species richness again, just to illustrate what I want to do next. So with any metric, we can quantify its change through time, put a regression line through it, get the slope, and it's managed through the graph, but we can also get the 95% confidence limits of that slope. And this next slide shows the change in the slope through time of these different metrics. So here we have, so anything that's 0 is a flat slope, it's not changing. These are 6 different metrics. We got richness, abundance, alpha diversity in the form of the Shannon-Simpson index, Q1, Q2, and beta diversity, a Jacquard, compositional change, just presence and absence, Morrissey-to-Horn, compositional change, taking account of abundance. So 6 metrics of biodiversity change, and I've shown them both for biomass and numerical abundance. So you can see here richness, well, it doesn't matter how we measure species importance, it's increasing, but because the 95% confidence limit is overlapping this line, it's not increasing significantly. Interestingly, the Shannon and Simpson index are absolutely flat on 0, pretty much. So there's no response there. And as you might expect from what I said before, there's a tendency for compositional change to be elevated relative to the 40-year. And you can see here that there's a slightly different signal for the beta diversity metric that takes species abundance into account when we use one biomass for numerical abundance. So just a quick mention about these 4 different spatial gills. They jointly account for most of the biomass numerical abundance in the system. So this is adding them together, they account for nearly 100% of species richness, most of the biomass, and almost all of the numerical abundance. So these 4 components of the system, 4 spatial gills, are really driving or controlling the diversity of the whole system. Now, to understand how these 4 gills separately and jointly contribute to biodiversity change, I use this method. So we have a metric through time, which might be species richness against time, and we plot the slope of the whole assemblage. Then we replot the slope after we've taken one of the gills and we have randomized its abundances through time. So for example, I might randomize the abundance of the pelagic gill through time, but keep everything else the same, and then compute the slope of change of the whole system through time. So change in assemblage slope when the abundances of species in the focal group are randomized across years. And that allows us to dissect out the impact of each of these groups on overall biodiversity change. So we can measure the effect size. So if the overall assemblage trend is reduced when the focal group is randomized, it means the focal group has the effect of pulling the overall biodiversity trend up. And conversely, if the overall biodiversity trend is elevated when this focal group is randomized, it has the opposite effect, it pulls it down. So that's a way of dissecting out the effect of these different components of the system. And this is what we find. So what is very interesting to me anyway, these are 4 different gills, soft benthic, approximate benthic, pelagic, and hard benthic. Think here in terms of richness. 3 of the groups are not changing in richness through time, but the soft benthic one, those are the guys that eat most of the crustaceans, they're actually contributing to increasing overall species richness through time. But that's being offset by the effect of the other ones, which are not changing. So there's a statistical averaging effect, a portfolio effect. If we look at abundance, this is just numerical abundance in this case, it's the pelagic ones which have the most dramatic effect in pulling abundance up. So different metrics are having different responses which are driven by different components of the system. And this is everything together, which is, I know it's a big, complicated slide, but the message I want you to take from this is the fact that the different gills are pulling the system in different directions, and they're doing so for different ways for different metrics. For example, here, you you remember, the Shannon index was one which didn't change at all. We have two of the gills contribute to pulling the system up. Two of the gills are contributing to pull the system down, so it averages out. Then if we look at what biomass is doing, it's also pulling the system in different ways. So these different components of the system are cancelling one another out in terms of their responses to biodiversity change. And this is telling us something about how these systems are constructed and how they are functioning. Now, it is interesting that we're seeing very different signals, or quite different signals, for the two measures of species importance, biomass and numerical abundance. And if we think about it, there's a tension between these two metrics. Biomass is a reflection of resource use, and numerical abundance is to a large extent a reflection of population dynamics. And obviously, both species have both characteristics, but there's a sort of tension between them. And that tension, to some extent, is mediated through body size. So I'll come to that in a second. So different temporal change responses amongst the spatial gills dampen biodiversity change at the average level, but ways for different metrics. And different responses between the abundance currencies may be related to shifts in body size over time. And we do see that in this system, the average body size of the species in the different spatial gills is changing over time. So this is the hard benthic gill, which is showing the biggest decrease in body size over time. And I won't go back to the slides, but that was one where there was this biggest difference between the responses in the different metrics between biomass and numerical abundance. And these changes in body size over time are actually quite widespread in many different systems. And this was a big compilation of data, again, based on the Biotime database. It's many taxa, but it's mostly fish, mostly marine fish, and it's showing that in many systems in the world, body sizes are beginning to decrease over time. So I realize there's a lot that I was covered in that section. What I'm going to do to finish is to try and tie some of these points together and think about the consequences of these changes for ecosystem functioning. And to do this, I'm going to leave the oceans again, but take you up the road to the Amazon, where I've been fortunate to work with some colleagues in the flooded forest. And this is a particularly nice data set which helps reveal some of these ideas in a little bit more detail. So this is the flooded forest of the Amazon, and it's Solimois part. It's Mamirawa Reserve, which is halfway between Manaus and Tabatinga, roughly. And my colleagues there have been surveying the fish communities in the flooded forest over a number of years. So we've got a data set for 3 separate years, 2003, 2012, and 2023. This is a very seasonal environment. The water levels can change by 12 meters. So we've got high water, low water, rising water, and falling water. And they've also surveyed 5 lakes, and have done it by surveying the fish associated with this floating vegetation. That's a data set. And this is what the habitat looks like. It's a very large, extensive habitat. It's heterogeneous in both space and time. So here again, we're finding a signal of a decrease in body size in these species over time. So in this slide, we've got the 4 seasons: high water, receding water, low water, and rising water. We've got the 3 years of the study with density plots, and this is the most recent year. And you can see that it's shifting to the left. So in the most recent year, in all the seasons, there's been a decrease in body size of species in this ecosystem. Interestingly, species richness has not gone down. If anything, it's gone up. So we're seeing this change in the types of species, the size of species found there, even though the overall properties of the system are in many ways very, very constant. These are just some ordinations to show how the system is changing through time. I've done it both for numerical abundance and biomass. I'll come back to that in a moment. So there are 12 points, so it's 4 seasons by 3 years. You can see that in the low water season, it separates out nicely from the other seasons. There's also a temporal change. This is the latest one. The system moves to the top left if we're thinking about season, and it moves to the right, thinking about the annual shifts. These are exactly the same data, that they're just plotted in the same. Well, this is the same data, they're just illustrated in different ways. Now, it seems that environmental variables are playing quite a strong role in driving these changes. And so I've overlain these ordinations with using GAMS, and this plots the minimum water level, and this plots the temperature. So these were the ones in the dry seasons up here. And you can see, I hope, that the water level is very low, but also the water temperature is very high. It's well over 30 degrees. And in the more recent years, it's actually increasing over time. So we can look at the structure of the system using these ordinations. We can relate that to the environmental variables that pertain in these systems. And we can also see which species are involved in moving the system from one state to another through time. So this again is the same ordination, but in this case, in numerical abundance and biomass, I have plotted vectors which show which species are most influential in moving the system from one point of the ordination to another. And you can see here that it roughly divides into species that do better in the warmer, drier years, warmer, drier seasons, and the ones that do better in slightly cooler, deeper water seasons. So we can identify which species are influential in shifting the composition of the system. I think this is my last data slide. So what we did was to set up a null model in which we asked whether the feeding groups of the influential species were more or less represented in these hotter and drier versus slightly cooler and wetter periods. So this slide shows the hot, very hot, dry seasons versus the slightly cooler, wetter seasons. It plots the feeding modes of the different fish: carnivore, detritivore, herbivore, insectivore, invertivore, and omnivore, I think. And so this is the null distribution, and the cases where the influential species are outside the null distribution are shown as colors. And what this slide is telling us is that if we're thinking about numerical abundance, in the hottest, driest season, omnivore species are overrepresented. If we're thinking about biomass, in the hotter, drier season, detritivore species are overrepresented. And then if we look to the other set of seasons, where it's, I always call it cooler, but it's really quite warm, we find the one thing that popped out there was that the carnivore species are doing much better in the seasons where it's not quite as hot and the water levels are higher. So we can see a shift in the composition of the species in relation to the seasonal change. Now, this is combined, of course, with shifts in body size. And so it's telling us that even though the overall properties of the system, the species richness, seem to be being maintained through time, the composition in terms of the functional roles of these species is changing. So there is a signal of reorganization in ecosystem function related to this compositional reorganization that we're seeing. And I guess, what should I say, that's the story so far. I think there are many big questions which many of you here are trying to answer about how ecosystem function is changing as a result of these biodiversity changes. And that's, I think, the big question for the future, which I hope many of you will have great success in tackling. So different functional groups of species are favored by different thermal conditions. What does it mean in terms of ecosystem function? That's your question. So ecological assemblages, including marine ecosystems, are undergoing rapid change. Biodiversity, as I've shown you, is being reshaped in complex ways. And a challenge for the future is to understand how these changes impact ecosystem resilience and ecosystem function. We don't understand that yet, but that is the big challenge. And to address that challenge, I think a way to proceed is to start to focus more on these co-adapted complexes of species and the extent to which they're supporting community resilience and ecosystem integrity. So let's keep thinking about the ecology and the biology of these species and the role that they're playing in protecting or making these systems resilient over time. At that point, I will say thank you very much to my many colleagues whose work I have presented here. Ecology and science in general are very much collaborative ventures, and the more we work together, the more we gain from one another. So it's a great privilege to be here, great pleasure, and thank you very much for inviting me.
Thanks, Annie, for this presentation and for putting the context that we are not a single compartment in terms of the marine environments, that we have complexity with other environments as well. So thank you for these other examples. Bueno, es el momento de las preguntas. Nosotros tuvimos un taller el lunes muy concurrido y las mejores preguntas fueron de los estudiantes. La tuvieron 1 hora entera preguntando y respondiendo. No tenemos 1 hora entera, pero tenemos al menos 10, 15 minutos de preguntas. Así que es el momento para levantar la manito.
Hi, good morning. I really enjoyed to see the integration of all the index. I am not a specialist in biodiversity, to have all together, that was nice. But when you start the presentation, you put 4 index in this kind of clock. And then at the end, you added the Jacker and the Morissette Horn index. So my question is, could be integrated in the same scale? And the second question is, I don't know the Morissette index. I understood is related to abundance, but I don't know what's the difference with the other.
Okay, so so the CHIVE framework does actually include both of those metrics. It's quite complicated, so it's too much to describe here. So the compositional change, you can look at it in terms of presence and absence of species. That is what the Jacquard measure does. But you can turn the dial as well, and so you become interested in the relative abundance, the relative dominance of different species. So the Morissette Horn index is one that takes that abundance structure into account. So I should have explained that more, but if you're interested, I refer you to the CHIVE papers. And I mean, she's absolutely marvelous statistician, but she can provide, or she has provided R code for all of these different methods. And she also has some very nice tutorials for them. So although the methods are complex, they're accessible. But anyway, well, thank you for your question.
¿Otra pregunta?
Hi, good morning. Thank you for the talk. It was amazing. I wanted to ask you, like, with all the research you have done through all these years, how and where do you think conservation efforts should be applied today?
That's a big question. I think, well, there are different ways of doing it. So there are two approaches in conservation. One is that you conserve species, like pandas. The other is that you conserve habitats or ecosystems. So I think that trying to conserve habitats and ecosystems is a good way, and doing it properly is a good way to try and approach conservation, because I think it's becoming clearer that these ecosystems are sort of a co-evolved, co-adapted complexes. So if you just try and save the panda without anything else, then it's like keeping it in a zoo. So I think it's important to try and conserve the whole system. Now, the other challenge, I think, is to try to move away from very naive understanding, just counting species. If I've said nothing today, apart from this, is that just counting species by itself is not enough. We need to think about the integrity, resilience, co-adaptation of these systems, because that seems to be what's promoting their ability to be sustained through time and to come back from perturbations. But that's a very difficult task. As I said the other day, there's a move in Britain towards biodiversity credits, which tend to be computed in an incredibly naive way. So how do we, as ecologists, make the case that biodiversity is a complex concept, and we need to find ways to protect all these different dimensions of it? I think that's a big challenge, and I don't know what the answer is, but I think, especially in today's world, when people want very simplistic answers, but I don't know. What do you think? You tell me.
Oh, you're asking. It's a very hard question.
You know as much as I do, so you tell me.
I agree with you in the fact that I think we should protect, like, not think about a single species, think about the whole ecosystem, all the system, and not focus just on one species alone. But honestly, I'm not sure about that. That's why I wanted to ask you that, because it's like a whole polemic thing to debate about. That's my opinion.
Yes, yeah.
Yo les dije que las mejores preguntas vienen de los estudiantes. Preguntas. Vamos, allí. Tomás, después.
Hello, Annie. Thank you for that great talk. My comment or my question comes related to what you were saying just recently. And I work on species interactions, and I always wonder why, in terms of biodiversity, species interactions are neglected or not considered as an important measure for, looking at the change in time and space. Yes,
I completely agree. So I'm going to ask you a question as well. Patricia, could you give him back the microphone for a second? Okay. So my question to you is, if we want to quantify species interactions, what measures should we use?
So, for example, body size is a good trait for species in order to infer species interactions.
Okay.
So there are new models that infer species interactions and the network of species interactions that are formed out of that. Body size is a really good measure to infer how species interacts, at least in terms of probability. You can say, okay, this species will interact more or less with this other one.
Yeah, I mean, the complication is if you go back to the work of Bob May and so on, species interactions are not uniform across the system. You have groups of species, as I showed for the Bristol Channel fish, that interact quite a lot with one another. And within those, it's probably the persistent core species that are doing most of the interactions, and the sort of rarer species that come in tend to be very low abundance, so they're not interacting hugely. They've got often random dynamics, so it's not that easy to find. We want to capture this co-evolution, co-adaptation, but trying to do it in a way that's easy to measure and ecologically meaningful. Not that easy.
Yeah, of course. Yeah, I agree. Thank you.
Maite.
Hi, Anne. Thank you very much for your talk. It's kind of a technical question maybe, but I wanted to ask you if you ever consider the variability within the life history of species, because I came across with this trait, intraspecific variability. And have you ever considered to do all your research? And if we integrate this approach, would you explore in terms of the number of individuals or in biomass?
I think that's interesting. I didn't talk about the Amazon work very much, but there are sort of really quite complex interactions between the numbers of individuals and biomass, and some of the environmental conditions, species with bigger biomass do better, and some species with smaller, more individuals do better. So there's a lot going on there. In terms of life histories, yes, of course. The challenge is always, are you doing a big community ecosystem approach, or are you doing a bottom-up species approach? So if you're doing a bottom-up approach, then it's often useful to try and focus more on individual life history variation within a species as well. Now, what I think the link might be is, for I said that in these systems, a small number of species are the ones that seem to be driving the compositional change. If we can identify those and dig more into the dynamics of those species, I think that might be quite interesting.
Thank you.
¿Preguntas? Ahí voy. Aguante. Okay.
Hi, thank you for your talk. Sorry if I missed it, but have you also considered homogenization signals, or that is communities getting similar, maybe with biotime, you can see that?
Yes, there's been quite a lot of work in that, and some systems are definitely being homogenized. So the one I showed to the west of Scotland, due to warming, there's a signal of homogenization there. And other places, they seem to be becoming more different through time. And also on Monday, I talked about some of our freshwater work in Trinidad, and there we had a survey with fish, invertebrates and diatoms. And I think the fish and the invertebrates were showing biotic homogenization across the different rivers in Trinidad, but the diatoms were showing biotic differentiation. So yes, people have looked at that, and it is another variable in this complex story. But there's still lots to be done with biotime, you know, so if you feel like having a look, please do.
Tienes ahí un tema, Fede. A trabajar, tienes tema. ¿Más preguntas? Por acá atrás. ¿No hay más? Thank you, Annie, for coming to Argentina with your own resources, for being here. We really appreciate your time, your experience, the holistic approach that you have given us this morning and on Monday as well. So thanks. This is part of the gifts that we have in present that we have for plenary speakers. And thank you so much for being here.
And thank you very much. And I wish everybody here, particularly the students, the very best for your careers in the future. Thank you.